Good evening, all of you. Today, you are engaged with the first session of the special webinar series, Insight into COVID-19, organized by the Society of Medical Laboratory Sciences, University of Peradeniya, to scientifically. You all know how terrible is this pandemic and how does it affect for each and every field in the world. Corona disease, usually we say COVID-19. It was identified as a pandemic by the World Health Organization. Anyone can be infected by breathing in the virus if you are near someone who has COVID-19 or by touching a contaminated surface and then your eyes, nose, or mouth. Therefore, the virus spread more easily indoors and in crowded settings.
As a latest update, around 217 million coronavirus cases are reported globally. Among them, more than 194 million people are recovered, while around 4 million deaths are reported. And also, according to the WHO reports up to 31st August 2021 about corona pandemic states in Sri Lanka, there are 436,081 total confirmed cases are reported with 8,991 deaths. As of 23rd August 2021, a total of 18,099,065 vaccine doses have been administered. However, Sri Lanka still struggles to move towards peace because the wave of this pandemic influenced adversely in our lives. We had to face different types mutations of this virus. The situation is getting worse day by day because now we are in lockdown situation and also around 200 deaths are reported per day. Don't know what the future holds.
So today we have invited Professor N.P. Sunil Chandra as the guest speaker of our program today. So on behalf of our society, we warmly welcome our guest speaker and senior lecturer, Dr. Charita Mendis, and all the participants to this program. From now on, you can engage with the session. Over the next few hours, you will be able to gain more knowledge about the history of coronavirus before COVID-19 emerges, how COVID-19 emerged and the early phases of pandemic, why COVID-19 is not yet settled down until today since it began of late 2019, mutation of the virus and how it occurs and what are the outcomes. And also, I would like to remind you, please kindly mute your mic while the session is going on.
Now, I would like to introduce our guest speaker, Professor N.P. Sunil Chandra. He is a virologist, category chair and senior professor of Department of Medical Microbiology, Faculty of Medicine, University of California. And also, he was a former chairman of Research Council, University of California from 2017 to 2020, and former president of Sri Lanka College of Microbiologists, 2006 to 2007. Apart from that, he holds the membership of Sri Lanka College of Veterinary Surgeons, Sri Lanka College of Microbiologists, and the National Academy of Science, Sri Lanka as well.
Good evening, all of you. I thank for inviting for this session as the guest speaker to talk about COVID-19. So your theme is Insight into COVID-19, Exploring and Understanding COVID-19 Pandemic. So my talk will be on coronaviruses before the pandemic, what coronaviruses we know, and also I talk about the origins of the coronavirus pandemic, as well as some aspects with regard to mutations and generation of variants. So First of all, I'll move on to the history of coronaviruses. Can you see the screen?
Yes, sir.
Yes, sir.
Right, the history of coronavirus. So we know this coronavirus pandemic or SARS coronavirus 2 emergence as a new thing. However, scientists, virologists knew coronaviruses long before. 1931, the first description of coronaviruses is reported. That is a coronavirus infecting chicken. We call it avian infectious bronchitis. It is quite devastating disease in chicken. Subsequently, in 1946, there was another coronavirus infecting pigs. We call it transmissible gastroenteritis. That again, a fatal coronavirus disease. In 1951, scientists found a coronavirus in mice. We call it murine hepatitis virus. What is the importance of having or learning about murine hepatitis virus? Because a small animal, laboratory animal, you can infect with this virus, coronavirus, and we can learn a lot of biology of coronaviruses. So most of the research done those days are very useful even today to study on the SARS coronavirus 2. Then in 1965, the first human coronavirus was discovered. And it was found as a labile virus from a schoolboy who had cold, a common cold, right? Now we call it common cold. And they found that coronavirus in this boy was similar to infectious bronchitis virus of chicken under the electron microscope. Then in 1966, several human coronaviruses, virus strains were recovered from medical students who had cold. So we named them as 229E, OC43, and OC38. At least some phylogenetic studies have shown one of those human coronaviruses, like OC43, have been originated from cattle. So it was earlier cattle virus and then jumped to human. Now we know, even after the SARS coronavirus 2, coronaviruses can move from bats to other wildlife species and then to humans. We call it zoonosis. Then from humans to back to the animals like tigers, in the case of SARS coronavirus 2, and pigs. The previously SARS virus was infecting pigs. So we call it reverse zoonosis. Okay, now we'll come back to human coronas of the coronavirus. It is about 100 to 150 nanometer, covered with an envelope consisting club-shaped, widely spaced surface projections. Now, this coronavirus we are talking today is severe acute respiratory syndrome coronavirus 2, SARS coronavirus 2, is the causative agent of COVID-19, the disease. First reported in Wuhan, China. It is in the Hubei province in late December 2019. So this is the seventh known coronavirus capable of infecting humans. So you can see there are 7 human coronaviruses we have today: human coronavirus 229E, human coronavirus 0 OC43, human coronavirus NL63, and HQ1. And then also we have MERS coronavirus, and the original SARS coronavirus. Of these, the first four, that is 229E, OC43, NL63, and HQ1 are common cold coronaviruses. They typically cause non-lethal, mild upper respiratory disease. However, MERS coronavirus and the SARS coronavirus and SARS coronavirus 2 can cause lethal respiratory disease. Okay. Earlier, previous slide, I talked about coronaviruses. We can talk something about coronavirus disease in animals. So usually when animals get coronavirus on their own, about 80% domestic species, including dogs, cats, cattle, pigs, they become seropositive, at least for one coronavirus in the first year of life. These coronaviruses can infect somatic, frequently epithelial cells of the digestive mucosa or respiratory tract. Because of this tropism, the disease syndromes are usually diarrhea or intestinal disorders. You can observe in bovine cows, and as well as respiratory syndromes, like common cold-like infection or deep infections like bronchopneumonia or pneumonia. Now I'll talk about mutations and recombinations of coronaviruses. What we know before the pandemic. Usually viruses compared to other microorganisms or maybe eukaryotes or bacteria, they don't have the regulatory mechanism, because when viruses replicate, there can be some errors in replication. The higher organisms can correct these or fix these copies, so they don't get any abnormalities usually. But viruses, they usually lack these regulatory mechanisms. Because of that, you can get variants. So the mutation rates also happen in larger magnitude in viruses. If you take RNA viruses like coronaviruses, it is even more compared to DNA viruses. Like for example, if you know smallpox virus was eradicated from the world because virus couldn't make variants, right? But RNA viruses can make, a lot of RNA viruses can make mutations and change, and so new variants emerge and become difficult to eradicate. Even if you compare with other RNA viruses, coronavirus RNA genome is longer than that of other RNA viruses. Therefore, the likelihood of replicating the copies and the errors can happen more than other RNA viruses. In addition to, they also have this recombination ability, recombination. So it is as high as 25%. So because of that, when RNA virus or coronavirus infect or emerge, it is not, initially it is not really adapted enough to survive with the host population, but because of the new variants can happen quite frequently in coronaviruses, there will be one variant which will become fit enough to survive in the or spread in the whole population. So that is why coronaviruses can cause adaptation to persist in the population. So what are the past experience before this pandemic? What we know about these mutations, recombinations, in the real situation. The scientists, when they studied murine hepatitis virus, they discovered deletion mutation of the S gene in this virus. And also they knew that by this mutation, there is alteration of the biological property in mice. And also in this murine hepatitis virus, there were recombinations they observed between temperature sensitive mutants. And also they noted these recombinations can occur in tissue culture, experimental animals, as well as in natural infections. So we know about these recombination and mutations of coronaviruses with our previous knowledge. And again, the coronavirus infecting pigs, we call it transmissible gastroenteritis virus. This virus had a deletion mutation somewhere in 1980s, resulted in formation of a new virus, coronavirus. We call it porcine respiratory coronavirus, and which became epizootic and causing a lot of deaths in swine industry. So that is one other example. By changing or having a deletion mutation in one virus strain like TG, we make a new virus, new coronavirus. That happened in 1980s in Europe. Again, even today we have this infectious bronchitis virus, even in Sri Lanka. New strains of IBV can arise in poultry flocks, and natural recombination has also been observed. So there are so many animal coronaviruses. Could these animal coronaviruses be transmitted to animals, to humans? Usually not. Once established a coronavirus adapted to species, they become usually contained within the species. Except few coronavirus strains which got broad host range. Example, SARS coronavirus, which was emerged in somewhere around 2002. So the virus can infect humans, civet cats, raccoon dogs, horseshoe bats, and swine. MERS coronavirus, humans, bats, hedgehogs, mammals. And bovine coronavirus, cattle, wild ruminants, camels, dogs, occasional humans. So there are few can infect humans, but other than that, the established coronaviruses in animals do not infect humans. But even infection doesn't mean the disease. Usually it is initially subclinical disease. Okay. What do you know about origin of SARS coronavirus? We believe SARS coronavirus originated in bats, SARS coronavirus 2. Usually SARS virus in 2002 originated in bat species. We call them horseshoe bats or genus Rhinolopus. They are the natural hosts. The palm civets are small carnivores. They are intermediate hosts and between the bat and the human. That's how it happened in SARS coronavirus. We also believe that origin of SARS coronavirus 2 also could have happened through bats, but we still don't know which was the first human who got SARS coronavirus 2, and also we don't know about the animal, right? But we believe with the other genetic studies on the beta coronavirus in bats, right? We found 96% genetic genome similarity. However, this bat coronavirus, the genetic similarity to the receptor binding protein, that is the receptor for the SARS coronavirus 2 is ACE2. The receptor binding protein has less similarity. So the bat coronavirus cannot infect humans, right? Although the genetic similarity is high. But at the same time, scientists found in China, Malayan pangolins, they have their pangolin coronavirus, which also not that similar to the genomic concordance. However, it has 99% similar to the receptor binding protein of SARS coronavirus 2. So the pangolin coronavirus can infect humans because it has the receptor binding protein. So therefore, it is believed recombination of the two viruses, so hybrid of the two viruses could have resulted in SARS coronavirus 2 that infected the first human in Wuhan. It probably is not Wuhan, maybe from other area, but the first humans would have been infected like that. How we classify coronaviruses? It belongs to the family Coronaviridae, and it has a large host range. So it has alpha coronaviruses, 4 major groups: beta coronaviruses, coronaviruses, and delta coronavirus. Now, today our concern is about beta coronaviruses. Mostly beta coronavirus represent groups also included the recently emerged coronaviruses, MERS coronavirus, SARS coronavirus 1, and SARS coronavirus 2. And also it includes human coronavirus, OC43, that is also one of the most prevalent infectious agents causing common cold in humans. Just something about the SARS coronavirus 2 genome and structure. So this virus is a positive sense single-stranded RNA virus. All these coronaviruses are positive sense single-stranded RNA viruses. It has the genome size of 30,000 bases containing 15 genes. So if you compare with the human genome, it's a double helix DNA, size of 3 billion bases containing 30,000 genes. Virus is very small. So therefore it replicate short time and also causing a lot of errors, and it cannot correct error, it make variants. The incubation period for COVID-19 usually 4 to 5 days, and most cases develop symptoms within 11 to 12 days of infection. If you discuss about case fatality rate, SARS coronavirus 2 about 3% or 3 to 6, which is lower than the SARS coronavirus, which has 11% case fatality, and MERS coronavirus about 34 to 37% case fatality. But we have now experience with SARS coronavirus 2 causing COVID-19, we see more fatalities. The deaths are more. Why we get more deaths? Although case fatality rate is low. That is due to the great number of infected cases. It is much greater than the other two, SARS coronavirus or MERS coronavirus. So the SARS coronavirus 2 infect many people in the globe. So that is why we see the large number of deaths, and we can feel it. How this virus is transmitted? The primary mode of transmission is by droplet spread, by person to person via inhalation. There are other indirect modes like contaminated hands or fomites. So this, you can see a person can sneeze or cough or loudly speak or talk. in certain can be more than 5 micrometer. It cannot travel, more than one in that is spreading can be direct. Okay. But in common cold, we usually see probably the same pattern with droplet infections. But the virus is mild infection, causing mild infection. I think we want to talk about early phase of the COVID-19 pandemic before discussing further how it happens. We all were just wondering, there is a new virus in Wuhan. This usually happened in somewhere around 17th November 2019 to 12th January 2020. We only found cases of infected persons in China. That is the Chinese that stage of infection, right? First period. Subsequently, we get East Asian explosion, that stage. That is 13th January 2020 to 1st February 2020, right? Like 3 weeks. So by that East Asian explosion of infection, virus was detected in 25 countries. That includes 13 Asian countries, including Sri Lanka, and also Australia, United Arab Emirates, Canada, USA, UK, France, Spain, Italy, Germany, Sweden. So it goes basically to Russia, Europe, and US. Outside China, it was considered as superclad B, that Asian super touching is East Asian explosion. Then you have a third period we call global pause in 2nd February 2020 to 19th February, right? So the virus that is from 20th February 2020 and beyond that, virus infection quickly turned into a global pandemic. So then European super type coronavirus, so superclad A was identified. The most European countries and Northeast USA, this virus spread among the population with high rate of spread, high incidence, mortality, and lethality. So those were the 4 main periods of early phase of the pandemic. Now today, you can see, I think you introduced with the today's review, the worldwide coronavirus by 31st August, we got 217 million cases and about 4.5 million deaths in the worldwide. In Sri Lanka, 36,000 cases and about almost 10,000, 8,991 deaths. So this is the situation. So it has become the global pandemic and large population has been infected, although less case fatality rate, the deaths are more. Why we find the greatest number of infected cases in the COVID-19? The past spread through the infected people within short period, coupled with modern travel, right? You see, when there was Spanish flu in 1918, a century ago, the traveling was very slow. It takes 6 months from one end to the other end of the world by ship, right? But here, within a day, you can travel around the globe. So this is one reason why it has spread worldwide. And also, there are other reasons. Main reason is asymptomatic infections. Up to 80% of the infected people are asymptomatic or mild disease which cannot be recognized. They travel or move around and infect others, and they don't know even they don't have the virus. So that reason, many people are infected within a short time. So this now usually scientists consider asymptomatic transmission is the Achilles heel of COVID-19 pandemic control. That is the weakness of this, despite the modern technology, we couldn't do anything because of these asymptomatic infections, right? You know what is Achilles heel, the mythological explanation for that, right? Okay. To support that, there are so many factors, viral factors. For example, mode of transmission is respiratory. There is need to have contacts. Like some other viruses, although deadly, they don't spread. Right? For example, HIV or rabies, they don't spread like that, right? But when there is a respiratory infection, from distance you can get it. Right? So that is one reason how it spreads so fast. And also droplets, micro droplets, leading to airborne infections. In addition, this SARS coronavirus 2 got this receptor binding protein, which can bind with the host receptor ACE2, which is abundant in respiratory and gastrointestinal tracts, as well as in several important vital organs. The receptor is there, so the virus can infect and damage, right? Also, the SARS coronavirus 2 has this mutation and recombination ability compared to other viruses. And that with the fast spread, and when the virus spread very fast in large number of people, new variants can generate quite frequently. Which can adapt more to spread in the population. Sometimes some become mild infections, some become severe infections. Now today we are experiencing a variant called Delta variant, which got high transmissibility, increased pathogenicity or hospitalizations compared to the original Wuhan virus. There are host factors as well as social or environmental factors resulted in infecting large number of people. One is having super spreaders. What are super spreaders? Some people from even beginning of the pandemic got high virus loads in their respiratory tract. So when they sneeze, more virus is released compared to others. They call super spreaders. Virus replicate more. And also there are super spreading events. For example, various concerts, weddings, restaurants, having meals without masks, riots, queues, fish markets, various super spreading events occurs, leading to infection by a large number of people. And also infection of the immunocompromised people can result in generating new variants. Because in these immunocompromised people, there are several rounds of replication of the virus, leading to generation of new variants. And also you have social factors, non-compliance with health precautions. Also people with underlying illnesses get more damage or deaths with the infection, because somebody with asymptomatic infection can transfer to a person who was susceptible. So gathering people, meetings, queues, offices, supermarkets are places where virus can be transmitted to others. Public transport, air travel, you notice initially, especially people traveling around the globe, especially in the flights under air conditions, virus is circulating. And so if one got infection, all others get it. Also, there are poor working conditions in certain working environments, such as crowding, poor ventilation, lack of personal protective equipment, such as face masks, sometimes shouting when the places are having more noise levels, then there is more generation of aerosols to infect them. Especially in fish markets, it happens, contact with other infected workers, poor hand hygiene practices, also inadequate or lack of cleaning of utensils. So all these things resulted in infection of large number of people and rapid spread within a short time. We talk more about SARS coronavirus 2 mutations. There had been several thousand variants identified, and all these variants have been identified and by various countries, and also the WHO make note of it. Right? There are 10 basic types belong to superclan A and B. WHO designated SARS coronavirus variants into two major variant groups based on their variants of concern, variants of interest, alert for further monitoring. Right? So if anything, any virus variant is under alert for further monitoring, if it becomes worse, then they put it under variants of interest. If it is even more worse, then it will consider that variant of concern. The viruses are grouped according to Pango lineage. Pango lineage means phylogenetic assignment of named global outbreak. So what are the outcomes of SARS coronavirus 2 emergent variants? What outcomes you will get? When a variant comes, it will have the ability to spread more quickly in people. Examples, alpha variant and the delta variant. It will got ability to cause either milder or more severe disease. Again, you have experience with alpha and delta variants. Virus also can develop ability to evade detection by specific viral diagnostic tests. For example, if mutation impacts one of the targets, the other RT-PCR targets will still work, but then those are the problems. Then again, decreased susceptibility to therapeutic agents such as monoclonal antibodies. Then ability to evade natural or vaccine-induced immunity. That is going to be the worst situation as an outcome if it happens with the variant in coming days. Okay, this is just the example of these variants of concern. B117, first reported in the United Kingdom, is considered labeled as alpha variant by the WHO. Then P1 variant reported in Brazil is considered it's labeled as beta. Then B1351 South Africa is labeled as gamma. The important one, the B16.72 is in India, we call it delta variant, right? Okay, now you have this problem. It is not the same enemy we are facing now. That enemy has changed from the initial pandemic virus. This is with regard to the delta variant. It has changed our calculations. So it's sometimes even guidelines for previous versions. Initially, you remember sometimes some of our health authorities as well as scientists used to say you don't need masks, but then you had to have masks. So all those things have changed, right? Over the time as we learn with the newly emerged virus. Not only that, it also changing further, like emergence of new variants. This virus is more contagious than the other known versions of the virus, other known versions of the SARS coronavirus 2, as well as most other viruses infecting humans. Delta variant is twice as contagious as the original Wuhan virus. This virus, the studies have shown 1,000 times or 1,000-fold higher virus content than the original version of the virus in infected persons. Virus in the airways of infected people 10-fold higher than the alpha variant. If you consider the contagiousness, Delta variant is twice as contagious as the original Wuhan virus. Virus can infect longer period and giving a lot more opportunity to pass the virus to others. That means more people get infected or transmitted. More severe disease than other known versions of the virus. For severe illness, or to be also, many people need to be hospitalized, many people require oxygen. So these are the changes. So our what we learn or our guidelines have to change with the aggressiveness of the Delta variant. Okay. It's just something I got it from the internet about Delta variant contagiousness. You see the virus is more transmissible than known versions of the coronavirus, as well as most other viruses like SARS coronavirus 1, MERS, Ebola, common cold, polio, bird flu, seasonal flu, as well as 1918 flu, as well as smallpox. So the Delta variant is more contagious. But it is contagious as the chickenpox. So it's one of the most contagious virus. So Delta variant is almost contagious as the chickenpox. Right. What will be the future of the pandemic? Future of the pandemic, what do you think about? So what is going to happen? That is something, some insights we should have. As most scientists believe, virologists believe, virus will most likely to become manageable, but persistent threat will be there. Virus can circulate for years to come, right? Hospitalizing and killing at least small number of people. This is what will happen, right? Same time, at the present situation in Sri Lanka, as well as many countries of the globe, coronavirus is changing rapidly, giving rise to new variants, right? And even more contagious, virulent variants may emerge because vaccination is not moving fast enough. Early in the pandemic, the target herd immunity, people thought that 60 to 70% of the population would help to control or get rid of the virus. But now, because of the new variants, that again pushed to at least 80%, right? So more people should be vaccinated. But even that, people think even that won't help, right? If in one country, if almost 80% or 90% people vaccinated, even more, it won't matter, because somewhere else in the world, some other country, or maybe developing country or poorest country, still not. From there, you will get more new variants and come back to the other countries. Right? This is the situation, right? So unless everybody in the world is protected, this cannot be controlled. Again, people think reaching the herd immunity may not matter at the end, because in the long term, there may be generations, the infection become tamed, or it will become something like common cold. Now we have the cousins of coronaviruses infecting humans. So when virus become like that, it will be one of another common cold virus. But it will take even generations, right? So that is the long-term outcome, what we'll get in common cold. But with that, we won't get herd immunity. So these are some insights with regard to the coronavirus pandemic. So we don't need to get this fair, right? This is, we have to change our attitudes. This is what will happen. But we should have the goal for vaccinating everybody and prevent fast spread to others. So if you prevent spread, then it will help not only to protect vulnerable people, but also prevent emergence of new virulent strains or variants. Right, that's all about my presentation. Thank you very much for your patience.
Thank you very much, sir, for the very informative explanation. Now we have reached to the end of today's session. Hope you got a better understanding of COVID-19 pandemic. So now, if you have any question to be clarified, you can either mute your mic and ask, or the chat box is also open for you. And I would like to remind you that we have sent a feedback form to the chat box. Kindly fill it out.
So is the vaccine virus is getting mutations, so it is getting mutated every day. So is there any possibility that our vaccines also induce another variant? Because if maybe in future, if a virus get a protection against our vaccine, then we have to get another doses, so invent another virus vaccine for that. So what is your idea, sir?
That's the idea. When we get these variants, even Delta variant or even further changes, it may not be effective that much. However, so far, these vaccines are effective to prevent serious illness or prevent hospitalizations. But I think United States and many countries, developed countries, thinking about giving a booster dose. And in addition, they are thinking about making the effect of new variants. So that will happen, right? So the vaccine will be coming as the booster, may protect for variants like Delta variant better than the existing vaccines. So these things will happen once everybody, actually the WHO telling, wait not to give the booster, not to introduce booster until everybody else get vaccine. However, some countries have already started vaccinating immunocompromised people with the third booster. So this is happened, but why the vaccine virus, there is no vaccine virus mutation, no changes, because this is only we use, even if you use live virus like vector vaccines, it only contains the genome, a gene, which spike protein gene, which is the one inducing the immunity. That virus is not replicating or making any change. Those are adenovirus vectors. So I think vaccine will be some other speaker might talk about. So there are inactivated vaccines and also vector vaccines like adenovirus vector vaccines as well as mRNA vaccines. They won't change, but the new vaccines will incorporate the new Delta variant or other variant changes, to cover the changes, the genome segments or genetic material.
Thank you, sir. So there's another question. I would like to know that what are the current researchers and clinical researchers that conducting with the coronavirus, related to coronavirus? I mean,
well, still actually
Sri Lanka, yes.
We don't know much about the virus so far. We only know how it has affected. But there's a lot of research going on with regard to antiviral drugs, with regard to immune mechanisms and pathogenesis of the virus. So those are the areas now people are talking. Every aspect has to be researched. Even the diagnostic methods should be researched. Or new techniques should be used, user-friendly techniques, especially with regard to the immunity as well as pathogenesis of the virus. Research has to be done, and a lot of people are researching on. I think if you look at various SARS coronavirus sites, you will see different research. But a lot of countries involved with the research. In Sri Lanka also, we should do research related to Sri Lankan requirements.
Okay. As it seems like there are no more questions to be clarified, therefore, we will wind up today's session. So this is the high time to pay our heartiest gratitude to our guest speaker, Professor N.P. Sunil Chandra, and to each and every person who contributed to make this event a success.
Thank you.